Hello, everyone, and welcome back to ANT Neuroeducational webinar series. My name is Farnoosh Safavi. I'm the head of education department at ANT Neuro, and I'm honored to be hosting today's webinar on TMS investigation of human motor cortex organization and reorganization with stroke recovery. This webinar will be presented by Dr. Dylan Edwards, who is the Nancy Wachtel Shrier Director of Jefferson Rehabilitation Research Institute, Director of the Human Motor Recovery Laboratory at the Institute, Professor in the Department of Rehabilitation Medicine at the Sidney Kimmel Medical College at Thomas Jefferson University, and the Adjunct Professor of Neuroscience and Director of the NeuroRehabilitation and Robotics Laboratory at the Edith Cowan University. He is dedicated to translating basic science findings in neurologically healthy people to develop and evaluate novel, clinically relevant approaches for neurological assessment and neurorehabilitation in stroke and spinal cord injury using non-invasive stimulation, neuromodulation, robotics, combinatorial therapies, neuroimaging, and telerehabilitation. After the scientific presentation, we'll watch a relevant demo prepared by my colleague Michael Matthews, our application specialist at ANT North America, who will walk us through the process of neuronavigated TMS. In the course of the presentation, you will also have the option to type your questions, which will be addressed at the end of the session as far as the time allows. So, Dr. Edwards, once again, we greatly appreciate you accepting our invitation and being with us here today. Without further ado, we're all ears and look forward to listening to your presentation. Thank you.
Wonderful. Thank you for the nice introduction, and it's a pleasure to be here and present to you today. Okay, so you can hear me okay?
Yes, we can hear you.
I can't see you. I'll take it as yes. I can hear you. Thank you. So I'm going to be presenting on transcranial magnetic stimulation using neuronavigation for the purpose of understanding organization of the human motor cortex in health and in disease after damage in the stroke, post-stroke, or spinal cord injury cases in the adult, and with recovery of upper extremity function. Now, I've been using transcranial magnetic stimulation for 30 years, actually. This month, in 1995, I finished my first research project mapping the primary motor cortex of skilled muscles in racket players, and I've over the next 30 years, I've continued this interest in probing the corticospinal system that's incredibly important for dexterous control in humans. But I transitioned to studying recovery of function about 15 years ago. And so that's really the basis of today's presentation, that I break into 4 sections. And I will weave throughout these sections as I'm going through my lecture, back and forward, but essentially these are the 4 areas. So I'm going to start with some background here. And I wanted to first define what we mean by motor mapping, and a working definition that I'll provide here, which is kind of represented in the cartoon, is assigning a cortical location to a quantified motor response following brain stimulation. So here in the cartoon, a very comical by Gary Larson from the far side, where the surgeons are, poking the brain and looking at which part of the body is responding to that poking. And it's not far different from the electrical stimulation by Penfield. So that's kind of the basis of what I'm talking about here, assigning a motor response in a limb to a region of the brain that is responsible for the driving that final common pathway to the muscle. Now, why is this important? Well, I like Penfield's homunculus here that you can see, and it's one of the most common drawings in neuroscience And many of us are familiar with this drawing back to 1948. And this was with the electrical stimulation of the exposed cortex. But recently, so in 2023, this Nature article came out questioning this original organization, or building on this original organization. And you can see here on the right, we have the proposed new representation of not just muscles, which is kind of called the effector zones, but these inter-effector zones, which are thought to represent actions and multiple muscles and act really like a premotor cortex to these effector zones. So they call these inter-effector areas. What's the importance of this? Well, I find that fascinating that so long after Penfield's initial drawings, we're revisiting the organization of one of the most studied areas of the brain. And I think that transcranial magnetic stimulation is a terrific tool, especially with the neuronavigation systems available that we're hearing about today, to probe this further, to understand structure-function relationship of the human brain. But let's dive back into the preclinical animal literature here, into an area that I'm really interested in, which is the recovery of function. And so this is a very famous study by Randy Nudo published in Science back in 1996. And I'll draw it. This is in the squirrel monkey, and this is in the healthy state. The dotted lines here represent the digit, so stimulating with intracortical microstimulation, we see responses in the digits. And then over on the right side, after injury to this effector or output region, and with therapy, with movement practice and recovery of function, this digit area moves to a new part of the cortex. So there's this reorganization or reassignment of the representation of those muscles. So this was, of course, indicating that there is plasticity in the brain, and that plasticity at least partly underlies the recovery of function. So this is with intracortical microstimulation in the squirrel monkey, but also we have more recent work by a colleague, Edmund Hollis, published in Nature Neuroscience in 2016, using a different method. So this is the mouse brain, and it's briefly it's showing here that with optogenetic stimulation, not intracortical microstimulation, they showed representations of which parts of the brain respond, which areas are responsible for forelimb and hindlimb movement, and then with damage to the at the level of the cord to the output zones for the forelimb area, you have, and with recovery of function and practice, you get a reassignment of this forelimb area to a new part of the cortex. So two separate studies using different techniques, using different animal models, speaking to the possibility of cortical reorganization that may relate to what happens in a human with recovery of function after damage at the level of the cord, or any part of the corticospinal system. And of course, in stroke that I'll talk about today, we can have damage in the cortex or in subcortical areas, or indeed in the cord, even with stroke. But any part of that corticospinal system that's damaged, we might, and with recovery of function, we might expect a reorganization. So people have studied this in humans. So this is a transcranial magnetic stimulation showing an experimental group that underwent this. It's of stroke patients that underwent this training called constraint-induced movement training. So it's a motor practice, very intensive motor practice, using the arm that's been affected by the stroke, and they have weakness and paresis, and a control group that also had stroke but didn't have the intensive intervention. And this paper claimed that the affected side here had an expansion of this motor map that was sustained for 4 months after the treatment and was related to the recovery of function that was not observed in the control group. You can see that the general morphology of these maps, they're not, they're quite irregular. And this is characteristic of these maps when probed by transcranial magnetic stimulation. Indeed, it seems quite similar to the intracortical microstimulation, although this is much less specific, of course, than microstimulation. So that's one thing to point out. So when you're trying to characterize these maps across many individuals, there's going to be really quite some variability in the shape and location. Okay. And so I've studied this myself with a group of patients using the NextStim system. It's a FDA-approved pre-surgical planning TMS device. And this is basically how the workflow is. So you have this grid, a 3x3 mil grid that's centered over the primary motor cortex with a biphasic current. You can see by these arrows, which indicates the direction of the current flow in the coil. And we systematically move around in these grid spaces to record where we're getting a response in the target muscles. So we're often measuring functionally relevant muscles of the upper limb. In this case, it was extensor digitorum communa, so which extends the fingers, and very important for clinical function in patients who have weakness of their hand. And so we mapped a series of patients, around 20 patients, and looked at how this map changed with recovery of function. And the hypothesis was that we might expect an expansion of the motor map that represents recovery of function, and the motor map might be reduced in size initially after in the early phases relative to the unaffected hemisphere, because it's representing the weakness that there and the damage to that pathway. And so this is a summary of the results. So you can find this paper on medRxiv, and it's under review at the moment at Transcranial Magnetic Stimulation Journal. And it's to really, for the purpose of this lecture, I wanted to point out two participants. So participant A had a subcortical ischemic stroke. That means that you won't see the lesion in the cortex, which you can see here. They had a Fugl-Meyer score of 38, which is out of a maximum possible of 66. So they had a reduction in their clinical ability, as shown on this impairment scale, that changed with time. So you have pre to post here. So they improve their clinical impairment. There were a reduction in impairment associated with an intervention that we did. And you can see this motor map. The black dots are everywhere that we stimulated, and the map, the color-coded map, shows really a fitted, smoothed representation of the responses that we see at discrete stimulation sites. And so you can see an expansion of this map that corresponds with increase in function. In participant B, who had a cortical lesion, and you can see the hole in the cortex here, we didn't observe such a change in map. Actually, it was quite strong to begin with. And they also recovered clinically. So the point here is that the jury is out on the extent to which these maps, reorganization of these maps or expansion of these maps, relates to recovery of function. And there's several reasons why that might occur. It could be that the timing was wrong and we didn't capture it in all subjects. It could be that it depends on where the lesion is located, in the cortex is implicated, and or if it's spared, as you see in the top patient. Or it could be that the sensitivity of the mapping itself is not high enough. And so it's on that point that I'll talk about our progression of where we are today. So let's talk about TMS mapping, and keep that last picture in mind of what I showed you, which is the prior art, I guess we would say now. Now, this group has had a presentation by Dr. Weiss on the Nature Protocols method, which was published in 2023. I did see there was a previous lecture at the ANT series, which is terrific. It's nice background for what we're doing here. And this, to me, is a major advancement in transcranial magnetic stimulation mapping. Very exciting. And where TMS can be used to localize cortical muscle representations on the primary motor cortex for an individual. And so you can see here this three-dimensional assignment of electric field on the motor cortex for a given muscle response. Relative to what I showed in the previous slide, which is really just a surface representation of where the center of the coil was, and an electric field estimation judging by the depth, estimated by the depth of the field. The electric field for TMS drops away very fast, and so that can be calculated. But now we're taking into account the individual's anatomy and how the electric fields might behave interacting with that anatomy. And so this is a major step forward, I think, for the field. And taking it one step further, Dr. Numenmar and colleagues are also applying this general methodology, but aiming to do it in a way that's clinically tractable by developing fast, accurate solvers. So instead of processing that takes many hours in only really quite experienced laboratories, this maybe can be applied in the clinic eventually, but they've made great steps towards that, because the calculations can be done in several seconds. And so in near real time, it's possible to be able to visualize the spread of the electric field and the density of the electric field in relation to the actual anatomy. And so this is really exciting for moving towards precision mapping and precision neuromodulation. The general workflow, for those who are interested, is you perform the anatomical MRI scan and break that the 3D anatomy into the different segments of tissue. You perform the TMS mapping for muscles of interest. We're very interested in proximal and distal muscles. And the intersection of the electric field map from the TMS onto the anatomy and the EMG response gives us, at any given location on the anatomy, an input-output curve. So that's the basic workflow of what we do. And you'll see the practical side of this in the demonstration shortly. This is how it actually looks in the lab. So here is Evgeny Kim, who came down from Boston, who's involved in this project as well. A patient who has a left hemisphere stroke that you can actually see here through a loss of brain tissue. And using the neuronavigation system from ANT and a MagPro stimulator, we applied this method, the Max Planck method, of many pulses at quite high intensity, 150% of resting motor threshold, and encompassing all of the motor map for the muscles of interest in the arm. And you can see that the one on the left here is actually quite posterior. We did indeed get responses here relative to the healthy or non-stroke hemisphere, which was much more kind of centered around or extending around the hand area. So that's the first thing to note. But once they have done the calculations for the electric field, we end up with an image like this. And if you think back to the prior image I'd shown, this is this is really transformative, I think, the calculations of the electric field distribution in the individual's anatomy, taking into account the gyral folding and the loss of tissue from, in this case, the stroke. And one interesting thing for this particular case, you can see the central sulcus is marked here in the dotted line, is that on the healthy hemisphere, contralateral to the lesion, it's kind of in the place where we would expect in the hand area because we were studying forearm muscles. And then interestingly, it's really quite posterior here on the postcentral gyrus. Now this is really interesting, and we wonder whether this is possibly because of the large extent of the lesion in this patient, or if this may happen more commonly, irrespective of where the size of the lesion, the location of the lesion. But now we have the tools to be able to investigate this, and indeed that's what we're doing, funded by the Chernowitz Medical Research Foundation, is to study patients with cortical and subcortical lesion and profile their the relationship of their clinical presentation to where the electric field can be mapped. Okay, taking that one step further, we're thinking about not only just the organization of the new state of the representation of the upper extremity after a stroke in the early stages and how that relates to their clinical deficit, but looking at how it changes with time, coming back to this, the preclinical studies that I showed at the start. So can this be a biomarker of change? What's the relationship of change in size or position of that representation as the muscle recovers function? And I think it seems to be an open question and probably differs depending on the nature of and extent of the lesion. So coming back to this diagram, I think that understanding the functional biology in M1 associated with recovery may assist in making predictions in recovery and response to rehabilitation interventions. And just a related note on that. So at the moment, there's an ongoing large-scale trial in the US called the Verified trial. And this is in part using the response to transcranial magnetic stimulation in the upper extremity to predict how people will recover following a stroke, which is very common and endemic across the world, and predicted to get worse with time. So it's a significant health problem. And it's very hard to predict how people will recover, and therefore in the management of patients, how to assign resources. So this is a really important trial, and it's relatively straightforward to do to assess a motor evoked potential in a patient as part of their overall medical workup and to help with their treatment planning. But also I'm very interested in guiding targeted neuromodulation. And we haven't had so much success. As you probably know, TMS is approved in medication refractory depression. So in psychiatric applications, it's an approved treatment. And we've tried to augment physical therapies post-stroke and also get this approved. But there's been a variable response to that. And in part, this might be because of the targeting might not be suitable at the moment. And so that's where this TMS mapping, I think, can come in as well. What's an example? This is an example of a motor training paradigm post-stroke that I've published with Dr. Kramer at UCLA, and we're poised to start a pivotal phase 3 trial. And it's an example of a treatment that a physical or occupational therapy type treatment of motor practice, where a patient with restricted function in their affected arm after their stroke can practice gross movements and dexterous movements in a gaming type environment in the home, supervised by a therapist with a webcam here. And they respond clinically over time, and we've published that in JAMA Neurology in a phase 2 trial, and now we're moving this to a phase 3 trial, and we're interested to understand if a therapy like this is associated with plasticity of the motor cortex, similar to what was seen in the squirrel monkey and the mouse. So the final part would be on prescribing the correct dose and location of neuromodulation. And I think this has a lot of potential, and I just want to explain an application of that. And before I do, I'll, for those who are not familiar with repetitive transcranial magnetic stimulation, it's one method of neuromodulation. So you can use various ways to modulate the central nervous system with electrical means or ultrasound and other methods. But non-invasive stimulation with transcranial magnetic stimulation, as I mentioned, is approved in psychiatric applications, and it means delivering pulses of TMS, not just single pulses as we do with the mapping. And depending on the frequency of those pulses, it has an after effect that can decrease the responsiveness of the cortex or increase the responsiveness. And it can be with consistent periodicity between pulses, or it can be with patterns of pulses, such as in the continuous state of burst stimulation or intermittent. And there's a recent paper on the mechanisms of how this works, proposed mechanisms. So that's neuromodulation, an enduring after effect to change the excitability of a region of interest in the brain. And you can imagine that if we had in a stroke patient, it might be difficult to understand where to target. Where would we target if we're trying to enhance the output of a particular brain region for a clinical benefit? And that's why these maps are so rich, and I think will be very informative for appropriate targeting and dosing. So by dosing, I mean estimating the electric field density in the region of interest for a neuromodulation effect. And I don't, it seems as if this has not been so precise in the past. And I think that this modeling can really help us with this, with correct dosing. So, but what this is in the case of where we have connectivity and we have motor evoked potentials in a reduced motor map or a displaced motor map, as you can see here. What happens if we have no motor response? What happens if there is a paralysis, a paralyzed muscle? What happens if we move off the motor cortex where we don't have MEPs and we're, say, in medication refractory depression, they target the dorsolateral prefrontal cortex. They use the motor evoked potential threshold to adjust the appropriate intensity for the individual, but perhaps we can get much more precision with dosing using this modeling method. So I'd like to present a case for you in spinal cord injury now of how we are moving towards using these kind of electric field models to help with prescribing the correct dose in a treatment for spinal cord injury. And in order to justify why we might do this in spinal cord injury, if we don't have a response, I wanted to present a preclinical study that I published with colleagues in New York at the Berkeley Neurological Institute, where TMS was applied to the rodent, and that there was a spinal cord injury model with a transection of the level of the cord. And you can see with this high frequency repetitive TMS, there's some growth of the axons through this lesion, which is very promising. So a neuron intrinsic reactivation of a growth program that closes off early in development for sprouting of axons that are damaged in spinal cord injury by treating the brain. And much of the interest has been in the past in spinal cord injury, neuromodulating the cord. So this is really exciting and another application of why we might want to have modeling based on this, the TMS mapping work that we're doing now to validate the models, that can be useful for clinical application for appropriate dosing in an area that people are not thinking about. So we're very excited about this spinal cord injury application, and we have funding at the moment from the Pennsylvania Department of Health. And here's an example of thinking of how to translate this animal study. So we have 2 coils as opposed to a circular coil targeting M1 bilaterally, and estimating the electric field in the region of interest. In this case, we're aiming to have around 80 volts per meter of electric field in M1 of the region, typically supplying the affected muscles in patients with traumatic spinal cord injury who have lost some function of their upper extremities, and it's often bilaterally. And then you can see the coils here. So we're positioning the coils using the ANT system with NeuroNavigation, and we are using the MagPro clinical trial coils, which are, they have a sham on one side and a real stimulation on the other, and it's very, you cannot tell the difference, and it's a blinded application. Let's see if we have, here it also shows the equipment. Very few people are doing this. I think this is quite novel, this bilateral stimulation with 2 figure-of-8 coils. So we're in the process of finalizing this protocol and beginning to apply it in patients. I have a short video here.
Grip a bit.
That video shows the frequency of stimulation and the duration of stimulation. This is just the experimental setup phase. We will have, once we identify the area with NeuroNavigation, we'll have rigid coil holders that hold the coils in the correct position, and we'll be recording the location of every pulse, so we understand precisely where the treatment was given. So that's, I guess, we've gone through a few areas now. This is how I'd like to wrap up and leave you with the idea that modeling with this sophistication and the precision that we have with the ANT system, with the individual's MRI scan, a high-resolution MRI scan, and with the great team that we have and the equipment that we have, is leading to state-of-the-art mapping of the human motor cortex in health to understand this homunculus, the structure-function relationship, and in disease after adult stroke, where we may have a large lesion of the cortex, and in spinal cord injury, where we also have reduced output to the affected muscles or no response in the affected muscles, which makes this the modeling even more important for correct dosing of the electric field in the brain. And I guess a final thought would be the potential to build in the robotic coil. So you'll see a demonstration in a moment of how the NeuroNavigation system works. And one of the nice things about NeuroNavigation is the experimenter, the person holding the coil, has real-time biofeedback of the coil position with 3 degrees of freedom. So it has the, the trajectory, the rotation of the coil, and the location of the coil in terms of the translation across the cortex. And this is rich information, it's rich biofeedback to improve the precision, as opposed to not using NeuroNavigation. And this is historically how it was done, actually, just with the scalp landmark with EEG coordinates. And and markings on the scalp or on a cap. And so the precision has been improved by giving this feedback to the experimenter, who can in real time visualize the amount of error for consistent targeting. But this image just shows if you have a robot device that can think about these 3 degrees of freedom with simultaneously, potentially with better accuracy and faster than a human. I think this has terrific potential for adding into the applications that I've described today for a single coil or 2 coils. And so I'd just like to leave you with that. There's many acknowledgements that I have for all of this work over the years, but I'd like to highlight you know, the current team who are helping with the TMS mapping and with application for using TMS as a biomarker and also for neuromodulation. And so I thank you for your time, and I think I'll finish here.